to talk about my work. I am giving a presentation today on Wikidata. I don't think there's too many presentations on Wikidata here today, but essentially I'm going to go into a project in the biological space. I'm a molecular biologist, and I work here in San Diego at the Scripps Research Institute, and we work on aggregating biological knowledge into structured databases that are machine readable, that really make searching through that data more efficient than searching through Putman publications and reading it yourself. And we also specialize in crowdsourcing, so these are the things I'm going to touch on essentially are linked data modeling, and how that aids biological research, and then how we can empower the crowd of biological researchers to add to the curation, to distribute the labor to the curation to biological researchers, because the current model is being overwhelmed with how much data we're generating in the biological research space. And I'm also going to present an application that I've built that works on top of Wikidata to present data to the researcher, as well as allows them to push information back to Wikidata. And I'll talk about what Wikidata is in a minute if you're not familiar with it. I know a lot of community still isn't. Real quick, I'm going to get my acknowledgments out of the way. As I said, work at Scripps. Andrew Sue is our principal investigator. Benjamin Good's our project lead. And then we have various contributors. Sebastian is in the room here, first scholar. Ginger is on our team as well in the back of the room. And essentially we're all working together from various institutions to make this project a reality where Wikidata becomes a central hub for biological knowledge. All right, so the issue that we're facing is, as I mentioned, the amount of data that's out there and how it's currently being curated. So more than 2 publications are published a minute, essentially, right now, and that rate is increasing every year as the research community grows. And the current model for curating this data, for taking the pieces of information that are in those publications and pushing them to databases where researchers can actually use a computer to find all the genes that have this relationship with this disease, is for professional curators to read the paper, identify the entities in there, link them to the database entry, and then push those entities to that database entry. And that's how it's done right now. And when you have this many papers being published every year, that is not a model that holds up, because it's really expensive and time-consuming to have people that are professionally paid to do this working around the clock doing this, and they just can't keep up. So essentially, we are trying to take data from, do everything we can programmatically, and pull data from different resources across the landscape of biological data. We have NCBI, which is the National Center for Biotechnology Information. UniProt is a protein database hosted by the European Bioinformatics Institute, the Swiss Institute of Bioinformatics, gene ontology, disease ontology, and then of course PubMed holds all these papers that hold all this information that researchers have gathered over decades. And we're trying to put what's appropriate into Wikidata to create a structured data model in there that allows the aggregation of this knowledge, as I mentioned, into a machine-readable format. And so we're taking the different interesting pieces from the different areas and putting them into a data model we've created. And that will serve as a place for other people to get involved and add information to. And so what is Wikidata, for those that aren't familiar with the project? It's about to have its 4th birthday. It's essentially a structured database, so it's the structured data what Wikipedia is to text. There's items in the database, much like articles in Wikipedia, but instead of paragraphs of text about the piece of information, it's structured data statements. So the city of London has population 10 million, and a qualifier on that statement would be as of 1999 or something like that. And then you can have various different statements for any kind of entity across any domain, just like Wikipedia. As I mentioned, cities, art, music, all that stuff is in Wikidata. And it was originally developed to host inter-wiki links between different language Wikipedias because they became inconsistent, so Wikimedia wanted a place that link the different Wikimedia articles or Wikipedia articles from different languages to each other, and Wikidata is first served to do that. And then our group, which hosts gene articles in Wikipedia, has used Wikidata to populate the info boxes. So on the right side, and I realize this is a very small screen for this kind of information, but on the right side of a Wikipedia article, we're probably all familiar with the info boxes. Our group originally started GeneWiki by creating gene stuff in Wikipedia and populating them with structured data in the info boxes. So as you can see here, there's a model of the protein structure is visible, and then various identifiers, expression patterns, things that are structured data, and this was all pushed to the Wikipedia markup. And it was, and when Wikidata came along, we changed the model to populate Wikidata with the information for these genes, and now a template pushes that information to Wikipedia article info boxes, which is much more consistent and can be pushed to any language Wikipedia as well. And so what we found right away is this can be useful for a lot more than just populating the info box on Wikipedia articles. This is a graph database, which has a lot of useful applications. And so we've put a lot more data in there than just human and mouse gene data. We now have in there the data for 109 bacterial species, as well as some other model organisms, organisms that essentially represent their species, that we've sequenced their genome and are heavily studied. And so we put all the genes and proteins for all these model organisms in there and link them all together in a structured data model. And this is just showing we get the data from the different places, put it in Wikidata, as I mentioned. And so this is the data model for bacteria. I came from a bacterial background in my research, and so my domain has been putting bacteria in there. And our gene and mouse and other work follows the same model, essentially, where everything is linked together. Bacteria is a Wikidata item, which has the class domain, essentially. Chlamydia trachomatis is a bacterial species that I used to work on as a graduate student. That's why I use it as an example here. But it's linked to bacteria through the taxonomic lineage. So genus, order, or family order, all of that gets you to bacteria. They're all linked together that way. And then the strain is linked to the species, so the individual bacteria from that species we're talking about, which has had its genome sequenced. And so then all the genes that came from that genome and all the proteins are also linked to that and linked to each other. And they're also linked to the diseases that they cause or they're associated with, the molecules that they interact with. So a protein can be an enzyme which interacts with molecules and catalyzes reactions. They're linked to all of those things in Wikidata. And so really it's a great way to find relationships between biological entities that we never would have looked at because they're not directly related. It's the same way Facebook suggests friends to you. They use a graph database to find connections that aren't direct. We're finding connections between diseases and drugs and genes that aren't direct. And then the idea is to present that to researchers that can then design experiments to look into those interactions and see if they're true in the lab. So essentially, this is great. This all lives in Wikidata. All this linked data. You can navigate Wikidata with a Sparkle query, which is a structured query language that most basic researchers don't know how to use, as well as most of the general public doesn't know how to use. So we created this data model in there, but it's really only useful to programmers at this point. So what we really need is applications that use Wikidata as their database, so that we can explore this data with a point-and-click environment. So that basic researchers who are trying to recruit to contribute to this graph, to this knowledge graph, essentially, where a piece is missing, we want a basic researcher that works on Chlamydia to see that there's a missing link in the graph and be able to add that themselves. So what we're doing is we're creating an application that allows them to do that. And so we're calling this the centralized model organism database. And it really is a pattern that we think is going to catch on. This could be an application for cities or art or music, using Wikidata as the backend. It doesn't have to just be Wikipedia. And essentially, what a researcher can do is go to our application, start typing the name of the organism they're interested in. If it's in Wikidata, they'll get a selection that they can choose from. They choose a bacteria. And this is really hard for you guys to see back there, I think. But essentially, it has the same front end, similar front end to something like NCBI's genome database, where you can look at a different gene, see the different curated attributes about that gene, scroll through the genome. This up here is a genome browser, and essentially it shows you where that gene sits on the genome if the genome was stretched out linearly. And you can scroll around, you can click on a gene, find out everything that we know about that gene that's been curated. And those types of annotations will show up in a box down here. Oh, sorry, this was the genome browser slide I was talking about. Essentially, that is a gene right there that sits on this Helicobacter pylori genome. All of this information is being pulled from Wikidata, which is a novel thing. Usually these exist in a private database on the NCBI servers or something like that. But this is all open, so anybody can add an annotation. It'll show up here in our application. So the different annotations associated with a gene. This is functional things that it's involved in. That, like as I mentioned before, professional curators can pull from a paper and push to a database that we then pull into Wikidata and display here. And the researcher can go and they can say, okay, that's interesting. This has ATP binding, nucleotide binding, ATPase activity. I don't know anything else about that gene, but they look up another gene and they say that there's no molecular function data available for that gene. And if they are someone that researches that organism, they can say, hey, I do know something about this gene. I study it, and we found this, and I have a paper I wrote right here that has this information in it. So what we've done is created an editing form. They just click on the button, add a molecular function, and they're presented with a form that allows them to put that information in to Wikidata. So essentially, they can find the term, and the science of this isn't really important right now. Really what I'm trying to get across is that applications can be created on top of Wikidata that allow people to both view the data and push data to Wikidata. But what we've done is created a form, and it's not done yet, this is the bare bones of it essentially, but they can find information in Wikidata that they want to link the protein that they're looking at to. Say it's a molecular function, as I mentioned. So they start typing and we query Wikidata and we find all the items that are the right kind of item, a gene ontology term that have that function. And essentially pull it from Wikidata, give them the option to choose the right one that comes from that paper, find the determination of that method, which was derived from an experiment, and then the PMID is important because then they can reference it with which paper they found it from. And then they can edit Wikidata, and this will implement O-authorization eventually, so someone can authorize our application to make edits with their Wikimedia credentials, just like the other Wikimedia tools. And then you get the message of successfully edited. You can look at the item you just edited in Wikidata. This is really hard to see, but it's down here. Extracellular matrix binding was just added to this protein item in Wikidata, and it was referenced from this paper in Wikidata, which is also a Wikidata item. So you can find all the pieces of information that have been referenced from that paper in Wikidata. And essentially, then it can be pushed back to the organism. And what you've done is you've connected that part of the graph to other parts of the graph, so that when people are querying Wikidata, they can say, give me all of the other organisms out there that create an enzyme that has this function. And so this is Spark, and I'm not going to go into detail on this at all. This is the query language I was talking about that allows you to say, give me all the items in Wikidata that have this molecular function, extracellular matrix, and give me the organism they came from. So you can create a short list of the different organisms right away that all have this same function, and that might be a great thing to inform your future research. The Mycobacterium tuberculosis also has this function that Helicobacter pylori does, but I'm going to study that pathway in Mycobacterium tuberculosis. And so essentially what we want to do eventually is create a query template where somebody pushes a button and this query is done for them. So we figure out what the proper queries are that people would be interested in and put those templates into the application, allowing people to use all of these back-end utilities that Wikidata has in a very easy-to-use interface that anybody can use. And essentially, this is what that query would look like. You have a function here in the center, and the organisms around the outside all have that function. And now, because you put that piece of data in there, all of these organisms are linked together. It's really powerful for exploring the data that we've already created and allowing people to add the missing pieces. And so with that, I'm done, and I'd be happy to answer any questions whenever question time is.
Again,